my pleasure to leave the stage to Sophia. Sophia Vinci-Booher, a postdoctoral fellow in Dr. Franco Pestilli lab in the Department of Psychology at the Brain and Science Indiana University. And she'll present us her work on the developmental differences in white matter tracts between and within the dorsal and the ventral stream. Please, Sophia, take it away.
Hi, so thank you for the introduction. I'll be presenting findings from a recent study that investigated the development of white matter contained within the dorsal and ventral streams, and also a white matter pathway that directly connects those two streams. It's widely accepted that visual information is processed along at least two pathways. There's the ventral and the dorsal visual pathways. We know quite a bit about white matter development in these two pathways. For example, we know that the tissue properties or the microstructure properties of this ventral stream here are adult-like by the age of 5, whereas the microstructural properties of this dorsal pathway undergo a more prolonged developmental trajectory, not reaching adult-like values until basically adulthood. So recently, a set of white matter tracts that connect the ventral and dorsal pathways have been rediscovered. So we are calling this the posterior vertical pathway. This posterior vertical pathway connects ventral and dorsal pathways directly. And we know essentially zero about the development of this pathway. So one of the goals of this work was really to understand more about the microstructural development of this posterior vertical pathway, because it has the ability to really communicate directly between these ventral and dorsal visual streams. We were particularly interested in understanding how the development of the posterior vertical pathway that I'll refer to as PVP, that I'll start using that terminology, how the PVP was related to the development of the dorsal and ventral white matter. The location of the PVP here seemed to suggest to us a particular feed-forward flow of sensory information. And there's a lot of work showing that sensory input or sensory flow is a major driver of developmental change. So we expected that this feed-forward flow might have some effect or some relationship to the order in which white matter tracts develop. So this model posits that some portion of the microstructural development is based on just receiving and carrying information. So the sensory information if sensory information were to follow the white matter architecture in a feed-forward fashion, it would first start out here in the occipital cortex along the vertical occipital fasciculus here. It would then travel down the ventral visual stream along the ventral horizontal pathway. And then it would travel up to the dorsal stream along this PVP pathway that we're particularly interested in here, before traveling along the dorsal horizontal pathway and then along pathways in frontal cortex, for example, the frontal aslant here that I'm depicting. So we started with a simple hypothesis. We hypothesized that the microstructure of the vertical pathway develops after the ventral pathway and before the dorsal pathway. And then we kind of tacked on the VOF and the FAT here to kind of get, to bookend the three pathways there. We collected diffusion MRI data from children ages 5 to 8 years and an adult comparison sample so that we could understand the relationship among the three pathways in development, but also in adulthood. And in brief, we first identified each tract and pathway using diffusion tractography. So here I'm showing you an example of a child and an example of an adult. And here are those same, the VOF, the ventral horizontal pathway, the PVP that we're very interested in, the DHP, and then the FAT here. For each tract, we computed the microstructural property, in this case, fractional anisotropy or FA, along each tract here. So here I'm showing you the ILF, and this is the tract. And we computed FA just along that tract here. So this is the mean for the adults. And this would be the mean for the children along the tract. We averaged across that tract profile to get the tract mean for the adults and the children. And then we averaged across tracts within a pathway to get the pathway mean. So this would be the mean for the the ventral stream, the ventral horizontal pathway. We did the same thing for the other pathways. So here's the dorsal horizontal pathway. That pathway is comprised of the SLF1 and 2 and the SLF3, for example. These are the same types of plots. And then here I'm showing you the means for that pathway with the adults and then children. All of these data were processed on brainlife.io using open web services, and these apps specifically. I'm not sure if this slide will be available after the talk, but you could definitely email me to ask for these specific links to the data so that you could reproduce the results if you like. All right, so we performed repeated measures de novo with 2 factors. The first factor was pathway here. So these are the pathways that I've been talking about. And then the second factor was age. We have adults and children. And the dependent variable was FA, based on the tract profiles approach that I just described. And we entered sex as a covariate of no interest. So this interaction was significant. The difference in fractional anisotropy between children and adults was dependent on pathway. And specifically, the age difference here, it increased linearly from the vertical occipital fasciculus onward to the frontal tracts here, in the way that we would have predicted if this feed-forward model of development has any sway or is correct at all. So the PVP here was the first tract to demonstrate a difference between children and adults, which is consistent with prior literature that has demonstrated that this ventral tracts here are adult-like by this age. So we decided to focus here more specifically. We wanted to look at how because the PVP was really the first tract that showed a developmental difference there. So just to find more evidence for this data-driven model, we expected that PVP microstructure should be more similar to, so that this microstructure should be more similar to the ventral horizontal than to the dorsal horizontal pathway. Because if this is mature, then it's sending mature signals through this pathway that can have a developmental effect. To look at that, we computed pairwise correlations among tracts and performed multidimensional scaling on the correlations followed by k-means clustering. So here, for example, this square is showing you the correlation between the FA and IFOF and FA and SLF1 and 2 in the child sample. And for the k-means clustering, we set k equals to 2 to test our hypothesis that the microstructure of this PVP pathway would be more similar to the ventral than the dorsal horizontal pathway. And this is what we found. You can see here, this is the first and second dimension from the MDS. And we find that the ventral stream does cluster, or I'm sorry, that the vertical pathway does cluster with the ventral stream and not the dorsal stream. So finally, we wanted to assess any relationship between behaviors and the microstructure of the PVP. To do this, we performed a multiple linear regression where we regressed composite measures of visual motor skill, fine motor skill, and literacy on FA in this posterior vertical pathway. And we found that PVP microstructure was predicted by visual motor skill. So as visual motor skill increases, so does FA in the PVP, and also by fine motor skill. As fine motor skill decreases, fractional anisotropy increases in the PVP. So our results not only provide support for our data-driven model of white matter development, but they also suggest a key role for the posterior vertical white matter pathways in the development of the dorsal stream. Our main findings are that the PVP microstructure is less adult-like than the VHP and more adult-like than DHP microstructure in 5 to 8-year-old children. We also found that PVP microstructure is more similar to VHP than DHP microstructure in children. And this PVP microstructure did correlate with behavioral measures of learning in children. So we interpret these results to suggest that the development of the dorsal stream trails behind the development of the posterior vertical tracts and may be driven by input received through those posterior vertical tracts during learning. So in the same way, we could say that the posterior vertical tract development trails behind the ventral stream development and may be driven by input received from these ventral stream tracts. These results in our interpretation are consistent with a large body of work that has demonstrated that sensory input is a major driver of developmental change in the brain. And they provide support for this feed-forward model of white matter development. White matter develops earlier in tracts that are closer to sensory input and later in tracts that are further from sensory input. Okay, so this is just my thank you and acknowledgement slide. So thank you to my postdoc advisor and all of my funding sources and all of the research assistants that's helped with data collection.
And thank you for the great talk. Very, very extremely interesting. I will be monitoring the Q&A. We have our 3 minutes with great timing.
Okay.
We also share the link to your website, to the lab website, so people will be able to check out the software. If I may start, I do have a question already.
Yeah, sure.
You mentioned having sex as a covariate in your analysis.
Mm-hmm.
And I wonder whether you did find any effect of that. Like, is there any difference in development between female and male?
Yeah, it's a great question. It's there's actually a lot of research showing that there are sex differences in white matter development, for sure. So it's a covariate that absolutely needs to be in there. That being said, in our sample, we actually didn't find any. It was not a significant predictor, and it didn't interact with any of the other predictors that we have in the models. Yeah.
And then I have another probably naive question that comes from my bias in studying language. I wonder whether you looked at differences between left and right, because we know that the development of the visual warframe area on the left side of the brain is, I wonder if that could play a role in.
Yeah, definitely. So we actually did do a hemispheric analysis of hemispheric cohesiveness, and we found actually that in the children there really wasn't any reason to separate left and right tracts, so we grouped them together. So that was true in the first 2 analyses. So in the ANOVA analysis, where we were looking at the developmental progression of tracts, and then also in the secondary analysis with the clustering, it didn't really matter if we looked at the left hemisphere, the right hemisphere, if we put them together, if we, it didn't seem to matter, but it did matter in the behavioral correlations. So when we really kind of dug deep into, I just presented sort of the big blanket result there. So that the result that I presented was that the posterior vertical pathway at large correlates with visual motor and fine motor skill, but not literacy, which was interesting to me. But when you kind of separate apart, which tract specifically was it, or was it the left hemisphere or the right hemisphere, or, it actually is, that correlation is driven by the left posterior arcuate being correlated with the visual motor composite score.
Interesting.
Yeah, so it showed up in behavior, but not necessarily in the developmental progression.
Right. Also, the other speakers, if they have questions, they can feel free to jump in or write in the chat. I do have one last from my side, a technical question. So you mentioned the software, and I confess I didn't check it out yet, but does it mean that the tracts are extracted like automatically? Or is the painstaking job brain by brain to extract the tract? And how this square between adults and kids, like, is it harder for?
Yeah, that's one of the strengths of the lab that I'm in right now. They this brainlife.io platform does a lot of things like that automatically that are, and it helps with reproducibility, so that somebody could just go, I didn't do it by hand. But a lot of these segmentation approaches that are on there and that are being used broadly have been validated against hand segmentations and things like that. But yeah, so it was automatic, and there was not, we did a lot of work to make sure that there was applicable to the children, and it seemed to be just fine. So yeah.
I will have to check it out, because I know my colleagues are trying to do DTI work on kids, and it's just difficult, and they never agree with each other.
Right. I mean, and it can be computationally intensive, all of these things, and BrainLife really does handle all of that very, very seamlessly. So yeah, I would definitely recommend it.
Thank you very much, Sophia. We have learned a lot.
Yeah.
So if there are